Welcome to Aquatic Biology. Today, we are diving into two of the most important concepts in water quality management: biochemical oxygen demand and chemical oxygen demand. If you have ever wondered how scientists know exactly how polluted a river or lake is, or how wastewater treatment plants ensure the water they release is safe for the environment, you are in the right place. We will build our understanding from the ground up, starting with how nature handles waste and moving to the clever chemical tricks we use to measure it. Let us get started. To understand how we measure pollution, imagine a massive feast is dumped into a small pond. Naturally, tiny bacteria in the water will immediately start eating this sudden abundance of waste. Just like us, these bacteria need to breathe oxygen to digest their food, grow, and survive. If there is a massive amount of waste, the bacteria multiply rapidly and consume a huge amount of oxygen from the water. This rapid consumption leaves very little oxygen for the fish, insects, and other aquatic life, eventually causing them to suffocate. This exact scenario is what we measure when we talk about biochemical oxygen demand. It tells us how much organic waste is in the water by measuring how much oxygen the bacteria use up to eat it. Now that we understand the basic idea, let us look at the formal scientific definition. Biochemical oxygen demand is defined as the amount of dissolved oxygen required by aerobic biological organisms to break down organic material present in a given water sample. Because temperature and time affect how fast bacteria eat, this measurement is typically taken at a specific controlled temperature, usually 20 degrees Celsius, over a specific time period, most commonly 5 days. We call this standard measurement the 5-day BOD. By measuring the oxygen levels before and after this 5-day period, scientists can determine exactly how much oxygen was consumed. A high value indicates highly polluted water, while a low value suggests clean water. To put this concept into practice, we use a straightforward mathematical formula. The formula calculates the oxygen consumed and adjusts for any dilution. The variable B represents the biochemical oxygen demand, measured in milligrams per liter. DI stands for the initial dissolved oxygen concentration, right after the sample is prepared. DF represents the final dissolved oxygen concentration, measured after the 5-day incubation period. Finally, P represents the volumetric fraction of the sample used, which is the volume of the actual wastewater divided by the total volume of the diluted mixture. If the initial oxygen is very high and the final oxygen is very low, the numerator becomes large, indicating a high oxygen demand and therefore high pollution. Let us see this formula in action with a concrete example. A scientist collects a water sample from a local stream. She measures the initial dissolved oxygen and finds it to be 8.0 milligrams per liter. After incubating the sample for 5 days, she measures it again and finds it has dropped to 3.0 milligrams per liter. Since the water was not highly polluted, she did not need to dilute the sample, meaning our dilution factor P is simply 1.0. We subtract the final oxygen level from the initial level to find that the bacteria consumed 5.0 milligrams per liter of oxygen. This moderate level indicates some organic pollution is present, but the stream is not severely contaminated. Now that we have solved a basic case, let us tackle a more extreme scenario involving raw sewage. Raw sewage is so polluted that bacteria would consume all the oxygen in mere hours if it were not diluted. A technician mixes 10 milliliters of raw sewage with pure water to make a total volume of 300 milliliters. First, we calculate the dilution factor P, which is 10 divided by 300, giving approximately 0.0333. The initial oxygen of this mixture is 9.0, and the final is 4.0. Subtracting these gives a depletion of 5.0 milligrams per liter in the diluted bottle. Dividing this by our dilution factor scales the result back up, revealing a massive final demand of 150 milligrams per liter for the raw sewage. Having calculated the values, let us visualize how this oxygen depletion happens over time. On the screen, you are looking at a BOD depletion curve. The horizontal axis represents time in days, and the vertical axis represents the amount of oxygen consumed in milligrams per liter. Notice how the curve starts at 0 and rises steeply during the first few days. This is when the bacteria are most active, feasting on the abundant organic waste. By day 5, marked clearly on the graph with a dashed line, the curve begins to flatten out. This 5-day mark is our standard measurement point. The graph shows us that while oxygen consumption continues slightly after 5 days, the vast majority of the biological activity has already occurred, making it a reliable snapshot of pollution levels. While the biological curve we just saw is the gold standard for assessing water quality, it brings us to a significant problem. Imagine you are managing a water treatment plant, and a toxic chemical spill happens upstream. You absolutely cannot wait 5 whole days to find out how bad the pollution is. You need answers immediately to protect the public. Furthermore, some industrial waste contains chemicals like plastics or pesticides that bacteria simply cannot eat. A biological test would completely miss these dangerous pollutants. To solve both the problem of time and the problem of toxic waste, scientists needed a different approach. They developed a method that uses harsh chemistry instead of hungry bacteria, which leads us to our next major topic. This brings us to chemical oxygen demand, or COD. Instead of waiting for microorganisms to slowly digest the waste, we use a powerful chemical to literally burn or oxidize everything in the water sample all at once. Chemical oxygen demand is a measurement of the total quantity of oxygen required to chemically oxidize all organic material in a water sample into carbon dioxide and water. This test uses a strong chemical oxidizing agent, typically potassium dichromate, mixed with boiling sulfuric acid. This aggressive mixture forces the oxidation of almost all organic compounds, regardless of whether they are biodegradable or non-biodegradable. Because it relies on a fast chemical reaction, this test can be completed in just about 2 hours, making it incredibly efficient for rapid water quality testing. To calculate this chemical demand, we use a specific titration formula. Let us break down what each part means. The variable C represents the chemical oxygen demand in milligrams per liter. VB represents the volume of the titrant used for a blank sample of pure water, while VS is the volume of titrant used for the actual polluted water sample. The symbol N stands for the normality of the titrant solution, indicating its chemical concentration. The number 8,000 is a conversion factor that represents the milli-equivalent weight of oxygen multiplied by 1,000 to convert the final answer into milligrams per liter. Finally, V is the volume of the polluted water sample tested. If the sample is highly polluted, it consumes more oxidizing chemical, making VS smaller and driving the overall COD value higher. Let us apply this formula to a real-world industrial scenario. An environmental chemist is testing wastewater from a textile factory. She takes a 50 milliliter sample. After boiling it with potassium dichromate, she titrates it with a solution that has a normality of 0.1. The polluted sample requires 15 milliliters of titrant, while a blank test with pure water requires 25 milliliters. First, we subtract the sample volume of 15 from the blank volume of 25, giving us a difference of 10 milliliters. This means 10 milliliters worth of the chemical was used up destroying the pollution. We multiply 10 by the normality, 0.1, giving 1.0. Multiplying by 8,000 gives 8,000. Finally, dividing by the sample volume of 50 gives a final chemical oxygen demand of 160 milligrams per liter. We have covered a lot of ground today, so let us summarize the most important ideas. First, biochemical oxygen demand is a biological test that mimics nature. It measures how much oxygen bacteria consume to break down biodegradable waste over 5 days. Second, chemical oxygen demand is the rapid, aggressive counterpart. By using strong chemicals, it oxidizes all organic matter in just 2 hours. Crucially, the chemical test captures toxic, non-biodegradable substances that bacteria cannot digest. Environmental engineers often measure both simultaneously. If the chemical demand is much higher than the biological demand, it warns us that the water contains toxic chemicals. Together, these two foundational metrics allow scientists to protect our aquatic ecosystems from the devastating effects of organic pollution. You now have a solid understanding of both concepts.